So for everyone listening, when you hear cellular reprogramming, maybe the Yamanaka factors, we're talking about that today. We are in that theme today. And just like we try and keep our bodies young and flexible, we want to keep our cells young and flexible as well. And that's what we are discussing with reversing DNA methylation of the epigenome. Because as you mentioned, Elke, I just want to highlight this as we age and because of intrinsic aging or because of extrinsic aging, like your exposure to UV radiation or smoke or other pollutants, stress, poor sleep, poor diet, you get these little marks on your epigenome that turn on and off genes, and those marks are called methylation. And so if we're able to reduce or remove some of these methyl groups, demethylate, then we can have our cells act younger, which is the whole purpose for both regenerative medicine in terms of acting younger, but then in terms of longevity, we want our cells to be in that young state for as long as possible to extend and prolong our skin span, just like we are with our health span. So just I know that's at a very basic level, Dr. Grönniger. I know that I'm, you can go into much more molecular detail than I am, but I think that just to give our audience some idea of what we're talking about now, I wanted to lay that foundation. And now that we have that foundation, let's talk about this compound that you discovered. I mean, it was well known in medicine, right? But you realized it had great potential for demethylating the epigenome. How did you do that?
Yeah, that's exactly how you described it. We are talking about dehydromyricetin or DHM. This is actually a compound which has been already, yeah, or has already a long history in the traditional Chinese medicine. It's known for its beneficial effects on the overall health. Like it's an oxidative compound. It's also inflammatory. It's really a very good compound, but nobody yet thought about DHM really being a rejuvenating active ingredient, and we also didn't do. I mean, when we started to look for interventions or to look for cosmetic compounds, we are first we first needed to set the status actually. So before we really start a screening for active ingredients, we need to build a kind of a solid scientific foundation, so that we need to understand what is really happening to characterize the specific epigenetic changes that are occurring upon age. I mean, you described it quite nicely, that we accumulate some DNA hypermethylation, but how this is really, how the dynamics are upon age. This is really needs to be understand quite well before you really can look for approaches or intervention strategies there. So this was the first thing we did, understanding what is changing and why it is maybe changing, and then looking for a specific target, because this is another one which you really need before you screen for compounds. You need to define a specific target, an enzyme or a protein that you can tackle with active ingredients. And once you validated that you really have the relevant target to introduce the DNA methylation changes you want to see in the end, then you can start to set up a drug discovery phase with the compound screening. And therefore, you systematically test large libraries with compounds and molecules, which in the best case really are a kind of a range about the whole chemical space you have. And in this kind of epigenetic screening we looked into more than 50,000 compounds in the end. And yeah, our outcome was definitely that still until now, we really haven't found a single molecule which is really outperforming dehydromyricetin. So it's really a unique and a powerful active ingredient we found in this sense.